Over the past decade, I've been at the forefront of developing a revolutionary technology called CRISPR that has come from the study of how bacteria fight viral infection.
This is a huge moment in science and also in medicine because CRISPR itself, this gene editing tool, was really only discovered almost a decade ago.
We are one little step closer in our quest for immortality as humans, and what you are about to watch is something that could potentially revolutionize the world of genetics and gene editing. This is all thanks to a tool that is so precise it can edit genes with near perfect accuracy. With the fusion of artificial intelligence and CRISPR technology, we now have a CRISPR gene editor for human cells that has been built from scratch. And if you are still wondering what this means for us, I must warn you at this point because it's gonna be a crazy ride from here. So, what is CRISPR? Let me quickly bring you up to speed with the backstory. Two scientists, Emmanuel Charpentier and Jennifer Doudna, won the Nobel Prize in Chemistry in 2020 for their work on this technology, also known as the genetic scissors. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. But how did this revolutionary technology come to be? Jennifer Doudna explains that CRISPR's roots trace back to the 1980s, when scientists first observed unusual repetitive DNA sequences in the genomes of bacteria. These sequences, later identified as CRISPR, were noted for their unique patterns of repetition and spacing, which led to the name CRISPR. Fast forward to the early 2000s, and researchers began to find out the true potential of these sequences. It was discovered that CRISPR sequences were part of an adaptive immune system in bacteria. Essentially, CRISPR acts as a form of bacterial immunity, allowing bacteria to remember and defend against viruses they had encountered before. In this system, bacterial DNA contains segments of viral DNA, known as spacers, which act as a memory of past infections. When a bacterium encounters a virus, it uses these spacers to guide an enzyme called Cas9 into the viral DNA, where Cas9 then makes a cut, neutralizing the threat. Do you get why it is called genetic scissors now? It literally cuts those viral threats. Both scientists figured there was something special here, so they introduced CRISPR-Cas9 as a tool for precise gene editing in other organisms.
CRISPR is amazing because it allows us to precisely edit the DNA in living organisms, including in people and plants. With CRISPR, we can change, remove, or replace the genes that govern the function of cells. This means that we now have the ability to use CRISPR like a word processor to find, cut, and paste text.
So, how does CRISPR-Cas9 work? The whole process starts with a piece of RNA, known as guide RNA, gRNA. This guide RNA directs the Cas9 enzyme to this precise location in the DNA. Once there, Cas9 acts like molecular scissors, making a double-strand break in the DNA. Hold on though, there's more. The cell's natural repair mechanisms then come into play. After Cas9 makes its cut, the cell can use one of two main repair processes. The first is non-homologous end joining, which can introduce small insertions or deletions. The second is homology-directed repair, which can be used to insert or correct specific genetic material if a template is provided.
CRISPR, amazingly, has already cured people of devastating disorders like sickle cell disease, and it's created rice plants that are resistant to both diseases and drought. Incredible, right? But the next world-changing advance with CRISPR will actually come from using it in a way that will allow us to go to the next level by editing genes beyond just in individual organisms.
With this new technology, we can now treat genetic disorders and save thousands of lives, which is incredible. Here's a case in point with Johnny Lubin, a sickle cell patient who happens to be one of the world's first beneficiaries of CRISPR.
2 years ago, it was hard to imagine 15-year-old Johnny Lubin doing this for very long. Born with sickle cell disease, an inherited disorder affecting the red blood cells, Johnny has been in and out of the hospital his entire life, dealing with bouts of extreme pain and other serious complications.
Part of a clinical trial for a completely new kind of treatment, Johnny is now one of the first people in the world to have his genes edited using CRISPR to treat his disease.
And I'm like, wow, that's pretty cool.
And scary.
And freaky.
The exciting thing is that Johnny has been been free of sickle cell pain crisis for about a year now. With this remarkable achievement, it's pretty easy to see why many of us are hyped up about CRISPR, but unfortunately, this technology has its downsides too. Despite its potential, CRISPR technology has its limitations. One of the most significant challenges is off-target effects. Think about it. Whenever you try to use your scissors, sometimes you cut good, other times, not so good. And on terrible days, you might end up almost cutting your finger. It's kind of the same thing with CRISPR in some way. Off-target effects happen when the CRISPR-Cas9 system makes unplanned changes to the DNA. While the technology is designed to target specific genetic sequences, there could be those rare cases where the Cas9 enzyme cuts at locations other than the intended target. These off-target effects can lead to unpredictable outcomes, including unwanted genetic mutations, which may have harmful consequences or introduce new problems. Think about this happening in the context of human gene editing, and you get how crazy it can be. Since humans have complex genomes, even small mistakes in targeting can lead to life-threatening issues. Another challenge is the precision of targeting. Although CRISPR-Cas9 is a powerful tool, it can sometimes struggle with very specific or complex genetic sequences. Certain regions of the genome may be difficult to target accurately due to their repetitive or highly similar nature. What makes the precision issue so tough is the fact that the CRISPR-Cas9 system relies on the guide RNA, gRNA, to direct the Cas9 enzyme to the correct location in the DNA. If the gRNA is not perfectly in sync with the target sequence, or if there are genetic variations in the target region, it could result in reduced efficiency and accuracy. Essentially, CRISPR is a coin with two sides. But how can we increase the odds of flipping the coin on the good side? This is where AI comes in. With machine learning and advanced algorithms, AI can analyze vast amounts of genetic data to identify patterns and make predictions that would be impossible for humans to do alone. AI's ability to process complex datasets and learn from them enables it to improve its own predictions over time. Microsoft's CRISPR.ml explains how the process can be carried out in weeks.
However, machine learning can dramatically accelerate this process. Working with CRISPR scientists, Microsoft created and trained a computational model that analyzed patterns and irregularities of DNA sequences and ranked the sites where CRISPR can be best applied. This allows scientists to more efficiently locate good sites for effective cutting, while also reducing side effects. Computing CRISPR-ML scores for the entire human genome would take over 200 years on a typical computer. But by using 17,000 Azure cloud machines, scientists could perform all their computations in weeks, enabling CRISPR to rapidly scale up and drive positive change globally.
AI can be used to design more accurate guide RNAs and improve the specificity of gene targeting with CRISPR, but that's not all. AI can predict potential off-target effects with precision by analyzing patterns in genetic data that might not be immediately clear through traditional methods. This helps in refining CRISPR systems to make sure that they only affect the intended genetic sequences. A 2023 scientific paper described how the use of deep learning helped to precisely control gene expression by accurately predicting the on- and off-target action of CRISPR tools. With AI's capacity to handle and interpret large datasets, it's no surprise that researchers are turning to it to advance CRISPR technology. The integration of AI into gene editing is a significant jump forward because of the potential to overcome some of the key limitations of current CRISPR systems and unlock new possibilities for genetic research and applications. New AI-designed CRISPR systems with large language models (LLM) are on the rise. Meta AI, through ESM Fold, is one of the technologies to take a stab at the tangled world of genetic sequences. The Transformer protein language model was trained with about 65 million protein sequences. The end result? ESM Fold was able to predict over 600 million protein sequences. Think about the potential in those combinations and figuring out what works to get the results you want. Here's another example. ProFluent, an AI startup, was able to develop a whole new gene editor using AI. They called it OpenCRISPR-1. This gene editor is made of a Cas9 type protein and guide RNA that were developed with the help of large language models. So how did they pull it off? First, they put together a dataset containing over 1 million CRISPR operons. Using this vast information, they built Open CRISPR-1. In their own words, "We demonstrated the world's first successful editing of the human genome using a gene editing system where every component is fully designed by AI." Essentially, they built an editor that learned from nature and then generated millions of other CRISPR-like proteins that do not occur naturally in nature. That is simply mind-blowing. Another example comes from the work done by a team from MIT's Computer Science and Artificial Intelligence Laboratory, CSAIL. In 2021, researchers led by Dr. Feng Zhang used AI to create a new CRISPR-based tool called prime editing. Unlike traditional CRISPR, prime editing is designed to make precise changes to the DNA without causing double-strand breaks. AI played a crucial role in optimizing the guide RNAs and editing mechanisms to enhance the precision of this novel tool. Talk about an upgrade. The prime editing approach, supported by AI, allows for the insertion, deletion, or correction of specific DNA sequences with a high degree of accuracy. Early trials of prime editing have shown promising results in correcting genetic mutations associated with diseases like sickle cell anemia and muscular dystrophy. This means we could have more success stories like Johnny's. So, what does this mean for us moving forward? The combination of CRISPR and AI opens new doors for treating genetic disorders. With higher accuracy, researchers can target and correct mutations more effectively, potentially offering cures for previously untreatable conditions. This advancement could be a game changer for personalized medicine. By tailoring gene edits more precisely, we can address individual genetic variations and improve patient outcomes. Second, it has applications in agriculture, where precision is crucial for developing crops with specific traits or improving livestock health. The improved CRISPR could lead to more sustainable and efficient agricultural practices. The use of AI in gene editing raises important ethical and regulatory questions. As we develop more powerful tools, it's important to ensure they are used responsibly and ethically. Those at the forefront of the technology know this too well, and so can tell us to be careful. These are 4 of the most important talking points when it comes to the application of CRISPR. Nobody wants to create Frankenstein's monster, and the only way to make sure of that is to handle things carefully. At the end of the day, we are probably still scratching the surface of what's possible with this technology. If you enjoyed this deep dive into AI and CRISPR, make sure to hit that like button and subscribe to stay up to date with the latest breakthroughs in technology and AI. If there are any other exciting topics that you want us to cover, be sure to let us know in the comments. And if you missed out on our last video on Neuralink and quantum computing, please be my guest.